In this last lecture, we'll recognize some diseases that are caused by bioterrorism agents and emerging pathogens. We'll be revisiting plague and smallpox, as well as learn about other smallpox-related viruses, viral hemorrhagic fevers such as Ebola and Marburg, which cause the type of bleeding that tends to be portrayed commonly in Hollywood outbreak dramas. We'll also look at an actual bioterrorist event that occurred in the United States involving anthrax. So, you may be wondering why we're covering these diseases, since many of them have not been reported in humans in Canada in the past decade, and smallpox has been eradicated. Due to their rarity, it is especially important to recognize the clinical features of these diseases, as well as the laboratory methods used to confirm their diagnosis, and recognize how the transmission of these diseases may be controlled and prevented, so that if these diseases occur either through acts of bioterrorism or natural emergence, they can be controlled through timely public health responses and interventions. As you can see from this table, all 4 diseases are caused by Category A bioterrorism agents. This is the highest priority group due to ease of dissemination as well as transmission from person to person. These diseases also have high mortality rates and the potential to have a major impact on public health. Therefore, it is necessary to prepare special public health responses to any reports of these diseases. Although we looked at plague in the first lecture on the history of communicable diseases, we're revisiting plague again because even though it doesn't cause large epidemics today, it's still present in some parts of North America, in the rodent population, and sporadic cases of human plague have been reported in the United States. The disease carried by fleas, plague, has an incubation period that can vary between 2 to 7 days. Initial symptoms may be flu-like, but then it may develop into different forms of plague. The most common form is bubonic plague. Lymph glands near the flea bite, usually in the groin area if the bites are on the leg, will become swollen, and they may even rupture. These swollen lymph glands are called buboes, and you can see a couple on the inner thighs of the person in the picture. More than half the cases will die if untreated. Another form of plague is septicemic plague, which is usually a progression from bubonic plague when the bacteria spreads to the bloodstream. Untreated cases die from endotoxic shock. The last form of plague is pneumonic plague, which is found in a small percentage of cases. This form of plague occurs when the lungs become infected, resulting in a cough with mucoid and bloody sputum. It is the most contagious form of plague and can be transmitted directly from person to person. It can also kill within days, and in many cases, parts of the body start to turn bluish-black, hence the term Black Death. This diagram shows different transmission cycles for plague. The most predominant cycle that we have today in North America is the sylvatic cycle, in which fleas become infected after feeding on an infected wild rodent. Humans become infected if they come into direct contact with either the infected animal or are bitten by the infected flea. The urban cycle is the one that occurred during the plague outbreaks in the Middle Ages, where domestic rodents were the main reservoirs. Humans became infected when they were exposed to infected rodents or bitten by infected fleas in their own homes. Pneumonic plague can result from either secondary spread from bubonic and septicemic plague or through direct contact with another human or feline cases of pneumonic plague. So don't let any cats cough in your face, just in case. So to summarize, infected rodent fleas carry the plague bacteria Yersinia pestis. Bubonic plague is the main form of plague reported in North America, and is usually transmitted to humans from wild rodents through flea bites. Bubonic plague is not normally transmitted from one person to another. On the other hand, pneumonic plague is usually transmitted directly from human to human. The primary reservoirs of plague are wild rodents, such as prairie dogs. Other wild animals, such as coyotes, are incidental hosts when they are exposed to wild rodents. Cats can also become infected with plague, especially when they catch and kill wild rodents, and as I mentioned before, can transmit pneumonic plague directly to humans. This includes both feral cats, which are cats that were once domesticated but have become wild after a few generations, and pet cats. Around the world, plague persists in large areas, especially where there are infected wild rodents. In North America, it is reported mainly in the western United States, in the area surrounded by the 4 states of Arizona, New Mexico, Colorado, and Utah, and this area is known as the Four Corners. The last reported human case of plague in Canada was in 1939. However, it is still a national notifiable disease today because it is a Category A bioterrorism agent. Therefore, all forms of plague are reportable. However, only pneumonic plague cases need to be reported to the World Health Organization. A case of plague is confirmed when there is a clinical evidence of illness together with the isolation of Yersinia pestis from body fluids or a significant, that is 4 times or greater, rise in serum antibody titers to Yersinia pestis fraction 1 antigen by enzyme immunoassay or passive hemagglutination inhibition titers. For laboratory diagnosis, different clinical specimens need to be submitted for different types of plague. For bubonic plague, aspirates from buboes are required, whereas blood specimens are required for septicemic plague and bronchial washings for pneumonic plague. Yersinia pestis is a gram-negative bacteria that belongs to the Enterobacteriaceae family, which includes gastrointestinal bacteria such as Salmonella, Shigella, and E. coli. When it's stained up, it has a bipolar staining pattern with more intense staining at both ends with a safety pin appearance. Immunofluorescence assays can also be used with antibodies to one of its antigens. In the flea vector, the bacteria multiply at room temperature and produce a set of virulence factors, which include a blood clot digesting enzyme called fibrinolysin and phospholipase. However, the bacteria also causes agglutination, which blocks the flea gut. This prevents the flea from feeding. The hungry flea keeps on biting and trying to get a blood meal. In doing so, it transmits more bacteria. In humans, at the higher body temperature, a new set of virulence factors called YOPs are produced. YOPs are the Yersinia outer proteins, which are cytotoxins that are injected into macrophages through the type 3 secretion system that is used by all Enterobacteriaceae. There are at least 7 different YOPs, most of which are enzymes. They kill host macrophages, and the dead macrophages fill lymph nodes, forming buboes. There are also a new set of virulence factors, the F1 capsular protein, which is present in the gel-like capsule of the bacteria and helps in phagocytic evasion, and a plasminogen activating protease, which enable the bacteria to adhere to host cells, facilitating entry and spread. Yersinia reaches the lymph nodes through the lymphatic system, and this results in bubonic plague. The bacteria can then spread through the bloodstream, resulting in septicemic plague, and this may be life-threatening as endotoxic shock can occur, caused by bacterial lipopolysaccharides. The bacteria can also infect other organs, for example the lungs, resulting in pneumonic plague. Plague can be treated with antibiotics, and if diagnosed and treated in time, death can be prevented in more than 90% of cases of bubonic plague. To prevent and control plague, rodents and fleas need to be controlled. This can be achieved by rat-proofing houses and storing garbage appropriately. Insecticides and repellents can be used for fleas. In plague endemic areas, pets should be treated for fleas. Wildlife and domesticated animals should be monitored for plague, especially rodents and cats. There are plague vaccines, but they only protect against bubonic plague and are not available in North America. Close contact of pneumonic plague cases should take antibiotics, as well as family members who share the same household with cases of bubonic plague. Families with human flea infestations should also be treated with insecticides, and hunters and veterinarians should use PPE, including gloves and masks, when handling infected cats. There are 3 different types of anthrax. The most common one, presenting in more than 95% of cases, is cutaneous anthrax. is acquired after contact with material containing anthrax endospores, for example, contaminated animal skins and tissue. Skin lesions form black scabs after 2 to 6 days. You can see one in the picture. A fifth of cases can become life-threatening if not treated. A rare type of anthrax is gastrointestinal anthrax. It is transmitted through the ingestion of undercooked food containing anthrax endospores. Symptoms include nausea, abdominal pain, bloody diarrhea, and ulcerative lesions in the gastrointestinal tract. It may become life-threatening in more than half of the cases. Last type of anthrax is inhalational anthrax. This is transmitted through the inhalation of endospores. Symptoms include a mild fever, coughing, chest pain at first. Then the bacteria enters the bloodstream, causing septic shock in 2 to 3 days, and then death within 24 hours. It is the most dangerous form of anthrax. More than 3/4 of cases will become life-threatening unless treated before the septicemic stages. Industrial processing of wool or bone from infected animals can generate endospores in poorly ventilated areas, so this is an occupational health risk. In Canada, anthrax is transmitted from infected animals. It is rarely from another human. Animals are infected when they eat plants contaminated with bacterial endospores. In 2000, there was an outbreak with more than 42 bison in Wood Buffalo National Park in Alberta. It is thought that the exposure in this case was through dust baths when the animals dug up the ground and rolled around in the contaminated dirt. Other outbreaks have occurred in herds in Northwest Territories, Saskatchewan, Manitoba, and Ontario. Epizootics are usually controlled through the mass vaccination of animals. No human cases have been reported in Ontario since electronic reporting began in 1991. In 2006, one cutaneous case occurred in Saskatchewan. He was a farmer, and there were a large number of dead animals on his farm, including livestock and wildlife, the majority of which were mainly cattle and bison. 36 neighboring farms had to be quarantined. Anthrax is on the A list of biological warfare agents. A confirmed case of anthrax is defined as a clinical case with laboratory confirmation of infection through the isolation of Bacillus anthracis from a clinical specimen or demonstration of Bacillus anthracis in a clinical specimen by immunofluorescence. Bacillus anthracis is a gram-positive rod which is encapsulated in spore forming. It is non-motile. These are some pictures of Bacillus. The one on the top is Bacillus anthracis, but the endospores is not present. In ENH222, there was a nice picture with the endospores staining up, which I've included, although this is not the anthrax bacteria, but another Bacillus species, Bacillus cereus, which can cause foodborne illness. Anthrax is a disease that has an occupational risk. Animals should be removed from anthrax-contaminated pastures and fields and the premise quarantined. Any dead animals should be disposed and contaminated material should be disinfected with formaldehyde or incinerated. Susceptible animals should be vaccinated and infected humans treated with antibiotics. In 2001, there was a deliberate distribution of anthrax through the U.S. Postal Service. 5 anthrax-laced letters were mailed to 3 media personnel and 2 Washington, D.C. senators. All were postmarked Trenton, New Jersey. This resulted in 11 cutaneous and 11 inhalational cases of anthrax, as well as 5 deaths, which were all inhalational cases. 35 postal facilities and commercial mailrooms were contaminated. 10,000 individuals had to be given prophylactic antibiotics, many of whom were postal workers. All the deceased had the Ames strain of anthrax. This strain was isolated from a cow in Texas and maintained at USAMRIID, which is the Infectious Disease Medical Research Institute of the U.S. Army. Advanced genetic testing traced the spores back to a single spore batch that was created and maintained by Dr. Bruce Iwins at USAMRIID. The FBI investigation concluded 10 years later on the 19th of February in 2010. The conclusions were that the late Dr. Bruce Iwins, who committed suicide acted alone in planning and executing the attacks. He had worked alone late at night in the lab where the spore batch was stored before the attacks and was one of the very few anthrax researchers nationwide with knowledge on how to create the highly purified spores used in the mailings. It was hard to imagine why a senior scientist who led the anthrax vaccine program would carry out such an unimaginable act of bioterrorism. He suffered from mental health problems and feared that his program would be discontinued. Other evidence included the use of envelopes that were all sold in a post office near Dr. Ivan's home and where he had a post office box. The letters were coded and had bolded A's and T's. Dr. Ivans was spotted throwing out a book on secret codes before the FBI searched his home. The anthrax spores were not weapons grade, but were exceptionally pure, similar to those used in vaccine preparations. There was also a heavily contaminated mailbox at the main entrance of Princeton University, where the letters were mailed, and this was associated with previous personal relationships of Dr. Ivans. Contents of email communication to close friends and to himself also provided additional hints and clues. If you receive a letter or package that has too much postage, a return address is missing or foreign, or your name or address is spelled incorrectly, your suspicions may be aroused. These usually are not the only signs. When the letter or package feels too heavy for its size, or wiring or powder can be felt inside, or the package may be dirty, stained, or leaking. There may also be too much tape or string, and the worst case scenario has a funny smell or sound such as a ticking noise coming from it. You may need to respond quickly. In these suspicious scenarios, it is best to remain calm. There may be nothing to it. However, it is best not to open the package and leave it where it is. Alert everyone around you to leave the room and close the door. Call 911 or the emergency response number in your area, then wait in a safe place till the police or fire crew arrive. In most circumstances, it is more common for letters or packages to be opened and finding unusual contents, such as a powder or liquid with a threatening note or something that is unexpected and cannot be identified. Once again, it is best to remain calm and leave the package where it is. Remove any items of clothing that may have been contaminated and seal it in a bag. Ask everyone to leave the room and close the door. Then wash your hands or take a shower with soap and water if possible. Call 911 or ask someone to call 911. Then wait in a safe place till police and fire crew arrive. Advice will be provided by emergency services as well as public health on following up the incident. All public health units have a bioterrorism unit, and we have had a few incidents of letters with suspicious white powders at Ryerson and also other parts of Canada.